So blotting techniques are very widely used analytical tool for specific identification of desired DNA and RNA fragments and the protein fragments from thousands of molecules. So it has vast scope in the field of molecular diagnostic. So blots are the techniques for transferring DNA, RNA, and proteins onto the carrier so that they can be separated and often follow the use of a gel electrophoresis. So these are the various techniques: southern blot, northern blot, and western blot. Southern is used for DNA separation or detection of DNA. Northern blot is for detection of RNA, while the western blot is for detection of protein. Now, southern blotting technique, it was developed by E.M. Southern in 1975. This technique is used to detect the presence of particular piece of DNA in a sample. So, DNA detected can be a single gene or it can be a part of large piece of DNA such as viral genome. So, steps of southern blotting. First is to digest the DNA with an appropriate restriction enzyme. So, DNA is being cut into small fragments. Further, these fragments are being separated based on their size and charge by using the electrophoretic technique. So, we can see that these are the different types of fragments separated onto the agar gel plate. So, this is the instrumentation of electrophoresis, and this is the capillary plotting apparatus. This is the plastic tray. This is the gel plate on which we have loaded the DNA fragments. Then this is a nitrocellulose plate. And over these, there are three sheets of filter paper. And over these, there are tissue paper sheets and then a glass plate and then few weights are being kept. So that what is the purpose of this assembly? It is just to transfer the DNA right from the gel plate onto the membrane, onto the nitrocellulose membrane. So these blots are being created onto the membrane. That's how it is called the blotting technique. So in the next step, what we'll do, these DNA fragments are doubly stranded, so they need to be treated with the alkali so that they can be denatured and converted up into the single stranded fragments. Further, we will use in the step 4, we will we will use this capillary blotting tray in which what in this assembly what we'll do, gel plate is put onto the tray, into the tray, and over this gel plate we will put a nitrocellulose membrane, and over this there are so many paper towels and paper sheets are there, then some weight is being kept. So the procedure preserves the distribution of fragments into the gel and creating a replica of gel onto the filter. So this replica means the single stranded DNA fragments are now transferred onto this nitrocellulose plate. Next, we will pour over this filter paper some known probes, and probes are actually, these are some radio labeled DNA sequences, and the sequences are known, so they act as an no single stranded DNA. These are the known sequences and radio labeled, so these probes can no be used to trace the desired DNA fragment based on to the complementarity. So on this nitrocellulose plate in the previous step, we will pour over the excess probe. So if the desired DNA, single stranded DNA is present, the probe will hybridize with it. Then further, the probe is washed away so that excess probe is removed from the filter plate. Finally, the plate is this membrane is auto radiographed, and which reveals the DNA fragments to which the probe is being hybridized. So this is how we can detect the desired DNA sequences are available in that given sample or not. So, southern blottings are used in gene discovery, mapping, evolution, and developmental study, diagnostic, and forensics also. It is also used for DNA fingerprinting and preparation of RFLPs map, and then also used for identification of transferred genes in the transgenic individual. One more thing I would like to add on to the probe. Nowadays, the probes are based on to the chemiluminescence, or there are some fluorescent material attached to these probes instead of radio labeled. So nowadays, these are frequently being used. Then northern blotting technique, that is a technique used for detection of specific RNA sequences. That technique is a little bit similar to the southern blotting, because here the single, instead of single strand of DNA, already we have the single stranded RNAs present, and which are being broke, no used. We will use the same restriction enzyme to break this RNA into small fragments. RNA is isolated from several biological samples, like various tissues and developmental stages of tissue. And because there is some drawback of this method, because RNA is more susceptible to degradation than DNA. Little bit contact with the RNA's molecule enzymes will lead to the complete degradation of the RNA. So stability issue is, of foremost concern in this northern blotting technique. So then samples are loaded onto the gel, and RNA samples are separated according to their size onto the agarose gel, resulting gel following after the electrophoresis run. So these are the spots. Further, this gel spots are being transferred onto the nitrocellulose plate using the same plotting a capillary plotting assembly. So all these single-stranded RNA is now being transferred onto the nitrocellulose plate. Further, this plate is treated with some known probes. DNA probes are being poured over it. So if desired sequences of RNA are available, so it will hybridize, and then extras of probe, just like southern blotting is being washed away. Further, this plate is being analyzed using the detectors, like if it is on based on the chemiluminescence, or it can be radio a labeling detector, like X-ray, using the X-ray technique. So, what are the advantages of this northern blotting? Now, the expression pattern of the sequence of interest in different samples can be compared. So, it is in standard for study of gene expression at level of messenger RNA, that is used for detection of messenger RNA transcript size, also used for to study RNA degradation. It can use to study the RNA splicing, RNA half-life, often used to confirm and check the transgenic and knockout mice, and also study if there is some RNA pathogen infection is there. Now move on to the Western blotting. Western blotting is used to detect the protein. This protein can be altered protein or pathogenic or toxic protein. So we can use the immunoblotting technique, which rely on a specificity of binding between a protein of interest and a probe. Here, probe is not a radio labeled DNA, but it is a labeled antibody which is raised against a particular protein. So, as to allow the detection of protein of interest in a mixture of many other similar molecules. So, what we have done, there are various steps. In the first step, the mixture of protein is now loaded onto the electrophoretic plate, and this is how separated. So these bands are being separated. Further, here there is a difference with that of southern and northern blotting. Like the instead of using the agar gel plate, we'll use here the sodium dodecyl lauryl sulfate amide gel. So this plate is being used, and there from the plate we will transfer these material onto the a nitrocellulose plate, same as in case of southern and northern blotting. Further, this plate is poured over with known antibodies which are against the particular kind of protein if it is present in the sample. So if the desired protein is there, so it will bind up with this protein. It is not bonding with this or that. It is specifically binding to this protein. So further, excess antibody is being washed away, and this complex is now retained onto the plate. further, this is the antibody one, and against this antibody we will use a second antibody, which is either radio labeled or it is labeled with some enzyme, which will manifest some enzyme reaction if the antigen antibody complex is being formed. So what we'll do over this complex, which is present onto the nitrocellulose plate, we will pour over the second antibody, which is labeled. So this is known as reporter enzyme. So second antibody will, wind up with the first antibody and antigen complex. So this is the like a sandwich ELISA method. So because the first antibody is sandwiched between the second antibody and the target protein, that's how this whole assembly is known as sandwich method. So if this kind of complex is being formed, so in enzymatic reaction happens, which reports us that the desired protein is available. So we can detect the target protein with the help of this Western blotting technique. What are the application? It is an confirmatory for HIV test. Then Western blot is also used in definitive test for bovine spongiform encephalopathy, in which the structurally altered kind of proteins are structurally as well as functionally altered protein are being formed in the brain. So it is being detected. Then third is some form of Lyme diseases testing employ the Western blotting. That's all about the blotting techniques. Thank you.